First of all, can everybody hear me okay?
Yeah. Yeah.
Okay.
Yep. I got you. Yep.
Good deal. This morning, Dr. Dave Dahlgren is joining us from Utah State University. Dr. Dahlgren is an extension associate who earned his master's degree and his PhD from Utah State University, working on the Parker Mountain population of sage grouse over there, and most recently spent some time over in prairie chicken country with Kansas Wildlife Parks and Tourism in Hays, Kansas, as a small game specialist over there, and has since returned to Utah State University, as I mentioned, as an extension associate. So we're super fortunate to have Dave on the line this morning, and good timing. This correlates pretty well with the recent release of a Science to Solution SGI publication that came out in January, and hopefully folks have seen that. That's entitled the Grazing and Sagebrush Treatment 25-Year Case Study in Utah. And that link was included in most of the hold dates and also the call-in information that was sent out, so you all should have access to that. And it is also posted on the SGI Science to Solution page. So today we're going to cover sage grouse and sagebrush management, and often this is a topic that probably generates a lot more questions than it does answers. But certainly I think something that is applicable range-wide and I think warrants as much discussion as we can really give it. So Dave's going to run through a presentation today, and then we're also going to open it up for questions. So I'd encourage you to take advantage of the opportunity to pick Dave's brain on the topic. And probably the easiest way to do that is to, in the webinar, maybe write your questions down in the chat, and we can go through those one at a time. And if you don't do that, we can also just reconvene here on the telephone line and ask questions as they come up here when Dave's done with his presentation. So just real quick housekeeping, if you please mute your phone, that would be really good. And just keep an eye out for future webinars. That schedule should be out and about, and I believe we have a staff call coming up in March for folks involved with the FGI SWAT. So with that, I'm going to turn it over to Dave. Dave, thanks so much for joining us this morning, and hopefully this will be a good deal and informative, and again, encourage some discussion here. So put your thinking caps on, folks, and don't be shy to participate. Thanks, Dave.
Appreciate it. Everybody can hear me okay? You're all muted, so you can't answer. I got it.
We can hear you.
Hey, okay, thanks. So I need to acknowledge a couple other folks, Dr. Eric Thacker and Dr. Terry Mesmer, that I work with in the Utah Community-Based Conservation Program. They're an integral part of some of the things we're going to be talking about today. Just as an outline for what we're going to talk about, we'll kind of talk an overview of the issue. I'm going to make some assumptions that I'm kind of preaching to the choir here as far as your knowledge of sage grouse, so we'll kind of hit some basics. I'm going to go over two different research projects today, and there's a lot of information here, so we'll try to get through it quickly, but I think there's some important things that we've learned about sagebrush treatment and grouse from these two projects. And then we'll kind of talk about some general principles, and then hopefully have some time for questions. So I do want to have some discussion. There's so much about this topic that we, like was mentioned before, there's more questions than answers, and a lot of very prescriptive ways of thinking about things. So first of all, sage grouse and sagebrush. Sage grouse and sagebrush. These are, as everybody knows, obligate to the sagebrush system, and sage grouse need sagebrush. So when we consider winter, we're talking 99.9% of the diet in the winter is sagebrush. In the breeding, for nesting, the research is explicit that the nests under sage are the most productive and used during the early brooding. And then in the late brooding, that's the one place where sagebrush cover becomes less important, and I think it's the place where we have the most room to discuss sagebrush treatment and removal especially in conjunction with benefiting livestock operations, which I'm going to assume is what most of you are dealing with, producers, and certainly what I deal with as part of my job. So let's talk a little bit about the ecology of sagebrush systems and what we know. I'm not going to go over everything, that would take too long, but there's this idea that, as you can see on the picture on the left, you have lower sagebrush cover, you have more grass and forb cover, more herbaceous. And the photo on the right, you have more sagebrush cover than you have less of the herbaceous. And this is a competition thing, right? A competition thing for resources. And this idea of sagebrush system leads us to the idea that if we remove the sagebrush cover, we get more forb and grass cover. And as I deal with producers, and I'm certain as all of you deal with producers, this is their prevailing philosophy when it comes to sagebrush landscapes. Certainly there's exceptions to that, but most of the producers I work with have this view. And it's not wholly wrong. We have research that supports this view, and I'm going to report some of that today. So it's certainly part of the picture. However, if you look at this picture on the left, here is a sagebrush where you have the interspaces are bare ground, and your forb and grasses are associated with your sagebrush cover. And we do see this as well. There's a great little publication, if you want to get your hands on it, from Extension here at USU, Should Ranchers Value Sagebrush and Why We Need Sagebrush. And it talks about some of this research that has shown this hydraulic lift that sagebrush can have in that soil profile and bring the soil moisture to the forbs and grasses and their root systems and access some of that deeper soil moisture. And so this is a facultative play that the sagebrush has in the system. And so this is also part of the picture, and we need to be thinking about it. The point here is sagebrush systems are more complicated. Like every ecological system I've ever looked into, it's more complicated than it is simple. And there's lots of relationships going on and lots of dependent factors. Are you in a high elevation area with deep soils, with more resources, there may be more competition versus a drier site with shallower soil where sagebrush may be actually facilitating your herbaceous. So it's something to consider as you move forward. And as we deal with agencies, and as I've heard from the NRCS, as I work with them here in Utah, really sagebrush treatments are a no-no right now in the sage grouse world. Most states have, I think Utah is a little bit behind on that. We were treating more sagebrush the last few years than most other states. But recently, and I just wanted to use this as an example, our Utah governor put out an executive order on sage grouse last year prior to the listing decision. And one of the items in that order has this wording: no state funding shall be approved for projects that materially eliminate sagebrush within SGMAs, which is our term for our priority areas, without consulting the Division of Wildlife Resources and a finding that such a project will have a net conservation gain for the species. And I think that last part is the most important part. How do we ensure a net conservation gain for the species if we're going to remove sagebrush for a sagebrush obligate? This is the crux of it all, and hopefully we can talk a little bit and come to some conclusion about that. So I'm going to start on Parker Mountain in south central Utah. You can see the inset there. We're down there kind of in the Capitol Reef, if you've been to Capitol Reef or Fish Lake area. The yellow outline is the border what I would call the Parker Mountain Study Area that we've been looking at. We have about 10 years of research down there. You're talking about, probably almost 300,000 acres of predominantly sagebrush. Just so you understand the area a little bit, the elevation moves from east to west. So it moves up in elevation east to west on this high elevation plateau, and then again from north to south. So you're moving from about 7,000 feet up to about 10,000 feet elevation. Everything west of that study area is Great Basin, and the study area and everything to the east is in the Colorado Plateau. This is a sagebrush semi-desert down there, so we're dealing with a lot more shrub cover and less herbaceous cover. And the experiment and stuff I'm going to talk about is predominantly happening in mountain big sage brush communities with some black sage on the hilltops, but we really focused in on the mountain big sage. And we're talking in that 9,500 to 10,000 foot range. So we're up high, and we get quite a bit of moisture on the place where we're going to be working and talking about here. So I want to talk a little bit about our experimental design. We really used this adaptive management approach where we started small and then we went bigger. So the little turquoise boxes are 100-acre plots that were put into a pasture, and those plots were randomly assigned treatment type. So they either were treated with a Dixie Harrow, a two-way treatment, a Lawson aerator, tebuthioron, or spike, or left control. So we had 4 replicates of each of those treatment types for control in those 16 plots. Okay, and then based on the results of that experiment, which started in 2000, and we published that kind of the short term, the first few years post-treatment, we published in the Wildlife Society Bulletin in 2006. So we moved from there based on those results, and we started management at a little bit higher elevation pastures, and you can see those pastures delineated there. We kind of started on the south end of this, so the green polygon, that's called south pasture, and then we moved north every year, a few years, and you can kind of see how those years of treatment are interspersed. The yellow are our reference areas, so those are untreated sagebrush stands, mountain big sagebrush stands, in those same pastures, okay? And so those were used for our control. So we did vegetation sampling randomly across our plots and across our pastures. We looked at shrub, grass, and forb cover. I'm not going to go into all the experimental design details, that would take too long. But we also looked at sage grouse use across these areas. So for the Parker Lake pasture, which is our 100-acre plot, we looked at pellet count in 2003 and 2004, and then we did pointing dog surveys in those areas from 2003 to 2009, and then we've also done pointing dog surveys in 2014 and 15 in the bigger areas, both in the treated and untreated areas, and I'm going to show you some of that data. The last part that I want to talk about is in our bigger pastures, those higher elevation pastures, we did clipping and weighing along transects. So we have forb and grass biomass, so basically forage, and we looked at shrub canopy cover in those areas, both in treated and untreated areas. So these are some of our results. I'm going to first address the Parker Lake pasture, basically our 100-acre plots, and kind of talk about that. And we're going to talk about the long term. This is data that a graduate student just got done analyzing. So in the top, this is shrub cover data, percent data on the y-axis, and you can see control in the top left, and then we kept that control data going through the other, compared to the other treatment types. So you can see shrub cover was reduced that first year post-treatment for Dixie Harrow, and then kind of started coming back, and it got close. And based on my experience of being there, you pretty much can't tell the difference anymore between where the Dixie Harrow was and where it wasn't. And you can see the picture in the very top right. That's the Dixie Harrow plot. That's an example of one that was treated. That's the first summer post-treatment. So it basically went down to dirt. Although we did leave, and this is important to know, we did leave intact sagebrush, both islands and larger areas nearby. You can see at the top of it. Loss and aerator, the shrub cover, basically within about 5-6 years, the shrub cover came back to control levels. It responded really quick. The shrub cover came back really quickly. The middle picture is an example of a loss and aerator treatment. It basically crushes the sagebrush, and Parker Mountain is very rocky. We actually, in one treatment time of doing this, we ruined the Division of Wildlife's loss and aerator. There was lots of rocks out there, and it banged it up pretty good. For the spikes, and you can see the tebutyron data in the bottom right, the shrub cover was reduced and then hasn't come back still. So the spike has a much longer lasting treatment effect. You can see the bottom right picture. The spike had more of a feathered treatment effect. And what I mean by that is one plant lived, one plant died, another partial plant died but lived, and then other plants just weren't even affected by the treatment. And so it really had this feathered mosaic effect. And that's what we were going for. The spike treatment was put on at a 0.3 active ingredient per acre. It was lower. We weren't out there to just decimate sagebrush. We really wanted this feathered effect going on. And that, I'm going to talk more about that here in a minute. This is the percent grass cover, and you can see control. And this is the same pattern. It's going to be this pattern of grass as we move in the future. So we had a little bit of grass response from Dixie right away, and then it kind of went away. Lawson took a little bit longer and then went away. And we really haven't seen a separation of grass from control with Lawson. The spiked areas, it took a little while, and then you see a response of grass a little bit later, and it stayed that way. We actually have some data from 2014, and it's still different. I just didn't report it here because it doesn't quite come all together, so we're just going to 2009. So this is forb cover now. So Dixie, not really a response from forb cover. Lawson, not really a response from forb cover. But the spikes, we saw a response from forb cover. There was kind of an initial spike, and then it's gone on and kind of stayed apart since almost gone in a different direction than the controls. And what's interesting about the forb response in the tebutyron is that we started to break it down a little bit more than just forbs. So this is what we call the facultative forbs, or more of the succulent forbs, these wetter forbs. And so we pulled those out of our data and just looked at the response of these more facultative forbs. And what you can see is not much in Dixie, not much in Lawson, but a stronger response in the tebutyron. And what we think is going on here is when you do a spike treatment, you're still left with a skeleton of a sagebrush, and that skeleton is able to hold snow cover, provide shade. It basically creates this more mesic microclimate, and that definitely affected the forbs. And so when we looked at these forbs across treatment types, the purple is more of the upland drier forbs, and the green and the kind of the blue-green, those are more of the wetter forbs. And you can see that the spike just had a lot more of these wetter forbs. It really is evidence that those microclimates, those wetter, more mesic microclimates were being created through that spike treatment. Even more interesting, we went back to the sagebrush or sage grouse literature and looked at all the different forb species that's reported to be used by sage grouse, and these are mostly crop analysis, so diet analysis. And when we looked at those forbs, you can see the Dixie it never fully separated from control, but was above control most of the years. Lawson, not much there. But the spike plots, those grouse forbs separated early and stayed apart the whole time. And what's interesting about this is when we look at our sage grouse use data, we get that same exact pattern. So this is pellet count. These are densities based on distance sampling on random transects. And you can see that our Dixies were higher than our Lawson or control, but not statistically different in density. But our spikes were statistically different from all of them. And there was just a lot more grouse using these spike plots, these 100-acre plots. When we looked at our bird dog data, same story, just a lot more grouse. And I don't have the graph up, but a lot more brood using our spike plot. Also, when we looked at our pellet, and we measured distance to edge, so either edge of a treatment or edge of a change in sagebrush type, like from black sage to big sage, and most of those pellets were within 30 meters, kind of. If you took on the whole, they would be within 60 meters of an edge. And that was not only pellets found in treatment areas, but pellets found in intact sagebrush. They were within about 60 meters of an edge, and most of them within about 30 meters of an edge. So while they liked those treatment areas, they still wanted that shrub cover nearby. And so that's so important to how we design treatments for sage grouse. Okay, so that's kind of the conclusion we came to from that Parker Lake pasture data, and then because spike was the most successful for the grouse and vegetation, we then started treating pastures a little bit higher in elevation, the same mountain big sagebrush areas, and just in different years now. Okay. So the data I'm going to go through now is not as much in an experimental design, but is more of an evaluation of that management action. So we took our vegetation data. So this is shrub cover, both live and dead, and forb and grass biomass. So we're not talking about forb and grass cover now. We're talking about biomass. And we did a PCA, which is a principal component analysis. And when we looked at the PCA, the red is the untreated transects, the black are the treated transects. So you can see they kind of partition out together. And when we look at the different pieces of data, basically the live shrub cover is associating with the untreated transects, and the dead shrub cover is associating with the treated transects, as well as an increase in forb and grass cover. So when we took a look across all the pastures and combined all that data, what we found was that there was definitely a response in forb and grass biomass in the treated areas. Okay, so that's kind of looking at all the pastures together. When we looked at the pastures each individually, so the lighter orange is forb biomass in treated transects, and the darker orange is forb biomass in untreated. And in all cases, the treated had more forb biomass than the untreated transects. However, none of them were significantly different statistically. When we looked at the grass biomass for forage, we had the same pattern, except now we had statistical significance in Nix, Forshey, and Chicken Springs pastures. What's interesting about this is Butte's pasture and South pasture were our shallowest soils, whereas Nix, Forshey, and Chicken Spring have much deeper soil profiles. And that's where we were getting the most bang for our buck, was where we had those deeper soil profiles. That's important when we're talking with producers and we're trying to mix grouse projects with livestock projects and how they come together. Those deeper soils allowed that bigger bang for our buck. When we looked at dog surveys in these bigger pastures. We did so in 2014 and 15, and you can see both years here. And there were some variants by year, how the grouse were using these pastures, especially in buttes. The first year there wasn't many grouse in 2014, but it had the most in 2015. Okay, mixed kind of had the opposite pattern. It's important to note, though, that buttes and mixed pasture are right next to each other. So these birds are using this landscape. But when we looked at all those treatment areas in the pastures compared to our reference areas that were untreated, by far more grouse were using our treated areas. And again, more broods were as well. So our data is showing us that the grouse are preferring these spike treatments. And again, we use that same active ingredient. So we were trying to get this feathered effect just at a bigger scale. And this is the data, how the grouse are responding to that. So what do we learn from Parker Mountain, if I can sum it up here? Spike was, when we compared it to our mechanical treatments, outperformed it both in the vegetation and grouse use. This feathered effect, this partial kill, I can't overemphasize that enough. When you have large treatments with 100% kill, you're on the edge of hurting the grouse and hurting this. It's this prescription of feathered effect and the small treatments that are going to be what help the grouse. And I like to think about it like this. So my grandma, she used to make chocolate chip cookies, and we called them chocolate chip surprise. And the reason we called them that was you were surprised when you found the chocolate chip. And that's kind of the principle that I would say, if you're going to do sagebrush treatment for grouse, it's a little bit here and a little bit there. It's not big blocks of treatment that's going to help. Okay. The spike helped create this mesic microclimate. We got more succulent forbs and more forbs that grouse prefer. The spike areas were preferred by grouse and specifically by broods. We increased forage. Again, the deeper soils, we had our bang for our buck. And this management, this adaptive management process, if you can do it and go through that learning process, what happened on Parker is the producers went with us through this process and have buy-in to the learning process. And that made a big difference in how that project and how that group is currently looking at future management. And lastly, again, late brood rearing habitat is the place that if sagebrush treatment is going to be good for grouse, this is where it's going to be. It's going to be in your late brood rearing habitat. The issue here is on Parker Mountain, the broods are moving up in elevation, and they're summering up at these highest elevations, while they lek at our lowest elevations and kind of nest in the middle. We do have nesting in these higher areas, but not as much as that middle area. And so because of that seasonal movement that this population goes through, we're able to do some of these treatments and say that we're targeting late brood rearing habitat. If our birds were using this area where these plots are for lekking and nesting and wintering, because there's lots of populations out there that use the same area for all these seasonal habitat types, our opportunity to be successful and help grouse with treatments would diminish if they were using these habitats for all their seasonal habitat needs. So you really need to know that about your population and understand how your treatments are fitting into these different seasonal habitats. And we'll talk a little bit more about that later. I'm going to move on to Deseret, and so this has to do with that Science to Solution publication that just came out. And we'll kind of go through this, and this is kind of at a bigger scale than what we were working on Parker. So this is in northern Utah. You can see the outline of the ranch there. That's about a 260,000 acre ranch. The BLN is the brown or tan, and you can see some BLN inholdings on the eastern side of the ranch, and then the BLN land to the north in Utah. I did not have the land ownership for Wyoming, but it's very similar to the north side where there's lots of BLN there on the Wyoming side of that line. So this is not semi-desert. This is Wyoming basin. It has a lot more grass and forb cover than where we were working down on Parker. So you can see this picture on the left. We're looking at data from 1989 to 2013. These are the leks in the area, and we separate them into kind of three different study areas. So there's the Rich, which is that north of the ranch in the circles, and that's what we're calling the Rich study area. The Western Wyoming is there. It's all the triangles, and then Deseret are the boxes. And so we separate them out like this. And what we basically said is we're going to look at these as three distinct breeding populations. Okay? Now, we understand that these three breeding populations are connected. Certainly during the winter, they're connected, and probably the same wintering population. But for the breeding, we kind of separate them like that once we hit that lekking season. So our approach was to look at the big picture lek counts and compare them across this landscape, then to look closer at brood counts, and then to look at, so Deseret had some treatments that took place, and so we want to look at sage grouse use within those treatments. Again, this is a case study. This is not a strong experimental design. It would be hard to implement such a design at this landscape level, and so we wanted to learn what we could learn through this case study. So the differences in management, DLL had high-intensity, low-duration grazing. It allowed them to rest about 30% of their pastures, and when they rested a pasture, they often got two full growing seasons based on their next rotation coming back in once it was rested. That rest is critical, I think, to these sagebrush systems. For Rich and Western Wyoming, we had either season-long or simple rotation grazing. For stocking rates, DLL had about half the stocking rate as Rich or Western Wyoming. For DLL, through this time period, about 15% of the sage grouse habitat had been treated. It averaged to about 1.5% of the habitat being treated per year. So there was kind of a regular treatment schedule on DLL within their sagebrush system. It's important to know DLL is not a sage grouse ranch. It's a livestock ranch with wildlife, a wildlife program. They do a lot of elk hunting. That's their main revenue for wildlife. For Rich County, there was less than 2% of that area that was treated, and for Western Wyoming, about 8% of the area. Now, when we say treated, we include wildfire, and that's what made up most of that 8% for Western Wyoming was two big wildfires. So when we looked at lek counts, at the first part of our period, all three populations are kind of trending together, and then in about 1995, it started separating. And what's happening there is the treatments on DLL began in the about 1993-94, the first treatments went in. So as those treatments started to accumulate, we saw this separation. And DLL about doubled their males per lek. And it remained that way for about 15 years. And then in 2010, we had a really bad winter up there. And not only did we have a bad winter, but come spring, we had lots of snow during the nesting period. We had snow. So we had hens on nests, and it was piling snow on top of us. That's not a good combination for a successful nest. That happened again in 2011. And so we saw this big crash, not just on DLL, but all the populations crashed together. Now, DLL's rate of decline is more, but when you have more birds, you're going to have a higher rate of decline as well. The graph on the right is just it's a Bayesian change point analysis. It's just looking at the probability that DLL's populations were real changes. And you can see the increase was real, and the decrease in 2010 was also real. We also looked at brood count. Now, I was not able to include these into the REM paper, and the reason is the methodologies for counting the broods were a little bit different, and so they wouldn't accept them as part of that paper. However, the counts took place at the same time of year in July and a little bit into early August, and no areas were double sampled. So I feel like the chicks per brood is still a decent thing to look at. And basically, it's this area here that the DLL wildlife manager went out and counted broods, and then a DWR biologist counted broods on this BLM area just north of DLL. Okay, and when we look at their brood sizes, DLL had about 4 chicks per brood on average, a little bit over that, and the BLM area had a little over 2 chicks per brood. So about twice the number of chicks on the DLL side in this time period from 97 to 2001. In my mind, and when you consider the importance of chick survival to sage grouse population dynamics, this matters, and in my mind was part of this picture of why DLL had such a response from their grouse population. So, sagebrush treatments on DLL. Again, they were putting in these treatments about 1.5% per year. It's important to note on this graph down here that most of the treatments were small. So they're in this less than 200 or 300 hectares. And here's an example in the picture. You can see this mosaic design, leaving sagebrush and taking sagebrush. Okay? So also, not only were the treatments smaller on the landscape, but they happened in higher elevations. So most of them were over 2,000 feet and into 2,100 meters, 2,000 meters and 2,100 meters elevation. What this really means is the basin big sage communities start kind of occur in this range here between 2,000 and 2,100 meters. Once you get over 2,100 meters, you transition from basin big sage to mountain big sage, and you increase in precipitation. Okay, and that's where most of those treatments were taking place. When we looked at the grouse use of these areas, we looked at broods in intact sagebrush, and we had reference areas that weren't treated, and then broods in treatment areas. And the number of broods in treatment areas far about doubled or more what we were finding in the intact sagebrush. Not only that, but when we looked at distance to edge, it's a really similar story to Parker in that most of those broods were within 60 meters of an edge. Okay, adults, we had a slightly more adults in treatment areas, but again, the adult birds that we found were using within about that 60 meters of an edge. So, from DLL, from our paper and this project, again, this is a case study, not an experimental design. So trying to tease out causal factors, mechanisms of why these changes is not really possible because of this case study format that we had to put things in. However, we had, based on the case study, we could say we had mixed results. We had this potential positive effect that we saw in the mid-90s that was maintained until we had that bad weather. And once the bad weather hit, DLL crashed, as did the other populations. And so there is a caution there that there's a chance, and we put this in the paper, we hypothesized that we may have treated too much sagebrush and started to hurt the birds by doing too much. We don't know that for sure, but it's a possibility. And when you look at sage grouse literature in other areas, once you start removing nesting habitat and breeding habitat, especially shrub cover, you hurt sage grouse populations. That's very well documented. However, few reported or published cases show an increase in sage grouse populations, and we were able to show that I think that's important. The story for most of the sage grouse world is they're declining, they're declining, they're declining. And I think it's important to say, where are they stable or increasing, and what can we learn from those kind of places? Again, that mosaic treatment pattern is important. I think once your treatment width goes beyond 120 meters, you're no longer benefiting grouse. And I would include that for your intact sagebrush areas too, that you want some room for the birds to get the cover that they need, as well as access to those forbs and grasses that are being increased in your treatment area. We have to throw out that caution that too much sagebrush treatment could be bad for grouse, especially when you're in those nesting zones. Okay, and I would include that for winter as well. In fact, in winter, it's really hard to justify sagebrush treatment in wintering habitat at all for sage grouse. We also have to think about legacy effects. So the data we reported in this study was data just from those years of 1989 to 2013. What we did report, but what happened on the landscape is large sagebrush removal projects were done in the 50s, 60s, 70s, and now a lot of those happened at lower elevations in our Wyoming sagebrush habitat in where the birds winter. And so the birds are wintering in crested wheatgrass stands that have some sagebrush that have come back in. But certainly those stands would be different if they had never been treated and would have more sagebrush cover in them. And so stuff that happened in the 60s could be hurting grouse now, with certain weather conditions and that kind of thing. You have to consider that. Sagebrush recovery needs to match treatment rate. So if you're in a Wyoming stand, it may be 30 or 40 years if you treat that before it comes back. And so your treatment rate on that spatial and temporal scales should be matching that, right? Whereas, like on Parker Mountain, we're getting brushed back within 10 years in our mechanical treatment. And so the treatment rate and scale at which we treat could change to match those recovery rates. And lastly, how is a treatment going to benefit sage grouse? It's probably through this nutritional pathway, right? You're increasing the forbs that are available to the grouse and possibly the insects that are associated with those forbs. That's this nutritional idea. But that has to be balanced against negative habitat or cover effects on the bird, and you need to be thinking about that. So let's talk a little bit about the guidelines that are still being used in our policy, both in federal and state agencies. And it's important to talk about kind of their recommendations because it matters when it comes to treating sagebrush. I think one of the issues we have with the guidelines is this. They're treated largely by the sage grouse conservation community as if they were handed down as the commandment. And they're not. They're not that at all. And we have to be careful with that as we move forward. In fact, the guidelines themselves, if you read them, you'll notice that they talk about the need for local information, and that if you have good local information on your populations and the habitats they're using, that should trump any data that they're purporting in the guidelines. And that idea is largely ignored in our policy and our resource management plans, and I think we need to do our best to look at the local data where we have it. So when it comes to treatment of sagebrush and the guidelines, when we're looking at different seasonal habitat types, this is what the guidelines say. In your breeding habitats, in your nesting and early brood rearing habitats, you need to maintain 80% of those habitats for a population. For brood rearing, and really what this refers to is late brood rearing, kind of that late June, July, August period, you need to maintain 40% of your habitats. And for winter, again, you're back up to maintaining 80%. So as you're designing projects, are we fitting spatially? Are we fitting within these, right? Now, it's important to know these numbers are the best projection, or I'm not going to say guess, but the best opinion of the experts that wrote the guidelines. There is no scientific evaluation that supports those percentages. It's not to my knowledge, it's not out there. And so we have to understand that we're still dealing inside these percentages with somebody's best guess. Okay? Now, for instance, my opinion, as working with sage grouse as long as I have now, I would want to maintain more than 40% of my brooding habitat. In fact, I would be up in the 60+% if they made me sage grouse king for a day and I could have my way. So just be thinking about that. I cannot overemphasize when you're dealing with sagebrush treatments and trying to justify treatment, you have to understand seasonal habitat types. Okay? So let's talk about how you might justify a sagebrush treatment for sage grouse. You know the limiting factor in your population, and could a treatment help address that limiting factor? So, for instance, if nest success and survival was your limiting factor, I can't see how you would justify treating sagebrush to help that situation out. However, if your limiting factor was chick survival and you had less than optimal form cover, then maybe treatment of sagebrush becomes an option. Okay? So think about that. Is form cover or diversity an issue in the place that you're working? If it's not, then maybe treatment shouldn't be considered, and you need to think about that. Can you define your population seasonal habitats? Can you draw those lines within the areas you're working? If you can't draw those lines, you're at risk of hurting the bird. And your probability of maximizing that certainty that you're going to help the birds is really gray. It's really shaded at that point if you cannot understand your population seasonal habitat use. And are they a migratory population? Are they migratory elevationally? Do they move between seasonal habitats? Or do they use the same area for their seasonal habitats? And if they do, which most of the populations I've worked with, there's at least some overlap between seasonal habitat types. That's where you got to be careful and very prescriptive on how you're going to do treatment. Can you focus on late brooding habitat? And if you can, if you know that there's certain areas the birds are going to, that for late brooding, that becomes, I think, the most justifiable place to do sagebrush treatment and increase forage for livestock. It's in that seasonal habitat type, and if that is separate from your breeding or wintering habitat. Can you ensure you're minimizing the effects on breeding and especially winter habitat? And you provide for a mosaic design. Now, as I work with producers, providing for a mosaic design is not the most efficient way to treat an area for sagebrush. It's just not. But there's a big difference between, let's say I have 10,000 acres of late brooding habitat, and so I'm going to treat 20% of it, so 2,000 acres. There's a big difference to the bird if you're going to treat a block of 2,000 acres or if you're going to do a big mosaic of 2,000 acres. There's going to be a huge difference to the bird based on the data that we have. So be thinking about that. I think this is an important question to ask yourself as you're helping the producers. And I understand the producers have their perspective, and we need to find mutually beneficial management practices, and that's how we're going to move sage grouse conservation forward. But we need to ask the question, what would happen if you left it alone? And is that the best thing for the bird? And because while you're there working with the producer, you're doing this for the bird, and we need to think about that. bottom line, can you maximize the probability of a positive impact to the grouse? That's your bottom line. I do want to talk about this one exception to winter habitat where I think we need to rethink things a little bit, and that's in wintering habitat where we've lost our perennial grasses and it's invaded by cheatgrass or Medusa head or some other non-native grass that's troublesome. Okay, so I'm talking about this right here. When we look at these kind of communities, and we have them here in Utah, I see them all over the place in the Great Basin especially. What are we going to do here? So if we're looking at the short term, you know, the 5-year mark, is this sage grouse winter habitat? Yes, it is. The probability of that being gone in a 5-year window is pretty low, that it's going to remain winter habitat for sage grouse. But let's go to 30 years. When I look at this right here, in 30 years, what's the probability that's going to still be there in 30 years? It's a lot lower in that 30-year window. And I think that's where we need to think about sage grouse conservation and what's the time scale that we want to look at. And I'm certainly more interested in that long term than the short term if we can balance those two. And we need restoration in this situation, and that's only going to take place through sagebrush treatment and getting a perennial grass back into the system. And it may, in this case, it would probably take some plateau to help you along the way to control your cheatgrass. And so, well, honestly, the science behind this kind of restoration of this kind of system isn't there yet. I don't think we really understand how to restore this, but you know, we're continually learning how to do that. But in this case is where I think we need to think a little bit outside the box of the guidelines, where you know we're thinking 20% and that kind of thing. And I think we need to think about some bigger landscape stuff to restore these kind of systems in the long term for sage grouse. And with that, I think we can take some questions.
That was great. Thanks so much. If we have any questions, you can either type them in the dialog box on the webinar or just go ahead and shout them out here. No, there's got to be questions. I'll call on people if I have to.
Yeah, this is Lars over in Oregon. I had a question. On the first part of your presentation, it seemed like something happened in 2005. A lot of those graphs seem to have a kind of a boost in production, both in your controls as well as your treatments. Just curious what happened in 2005.
Yeah, that was a wet year. We had a good snowpack and a pretty good spring. 2006 was a much drier year. So, you know, that understory is going to change based on your annual precip. But like, and you pointed out, the control responded as well. And so, I mean, that's our comparison, right, is that in reference to the control.
Hey, Dave, this is Taylor.
Hey, Taylor.
I was wondering if, so we've seen some preferential use by livestock using our GPS collars on Deseret. I was wondering in the future if that information will be found, that the sage grouse will be using those treatment areas in the future like the cows have been seen to do, and if that'll be sort of like an amendment with this paper in our grazing studies and show the correlation between the two.
Yeah, I think you're right, Taylor. I think so we have, Taylor knows this, not everybody else knows this. We have radio marked grouse with GPS on Deseret right now, so we're continuing to monitor. We've also had VHF on birds on DLL for the last 4 years. So yeah, we will look into that in the future. If they're doing what the rest of the birds that we've monitored, it'll show a preference for treatment areas. and I would expect that. The other update to that story, the DLL story in that paper, would be what's not in those LEC count graphs is what's happened the last 3 years, since about 2012, well, 4 years now, and that's a major upswing in the population. So as that population continues to grow in Rich, are the DLL LECs going to grow at a faster rate than the other two study areas? And that would be that's my question based on the past, and we can look at that in the future. Right now, I would expect that based on what we've seen in the past.
A couple other questions there in the chat, Dave. I don't know if you can see those or not.
Yeah, I got them. So, any thoughts on research on fire effects compared to other treatments? We did on the, well, I shouldn't say that. So on Parker, we did not do any fire stuff. On DLL, there was some small treatments with fire at higher elevations, but they were not monitored with bird dogs for the grouse count. So we don't understand that for DLL. I was part of a project on Anthro Mountain out in kind of eastern Utah, where we used fire, and this is part of Dr. Thacker's dissertation, and he showed stronger use, more use by broods and grouse in general on small mosaic fires in that area. So those fires mimicked kind of what we saw in the spike treatments and that kind of thing, rather than a large scale fire. So they really designed them for that more mosaic. I think the only place you can really talk about fire for grouse right now, based on the research we have, is in those higher elevation mountain big sagebrush zones, which Anthro was. I think if you're gonna talk about fire in Wyoming sagebrush, you're gonna have a hard time justifying that for grouse, and there's research out there showing that. Any other thing about fire that we're on it? Okay. The next question, in high elevation mountain sagebrush sites that are not in sage grouse winter range, what can or should be done in areas of dense sagebrush with little to no remaining grass and forb understory? Would you need to reseed following treatment, or would it be best to not treat these areas at all? That's exactly the case that we had on Parker Mountain. So we had non-wintering habitat at high elevations with very dense mountain sagebrush canopy cover. And I didn't mention this, but our pre-treatment cover for mountain big sagebrush was in that 30 to 40%, which is very high. And we assumed was limiting that grass and forb understory. What's interesting is we did not reseed after spike. So we had enough seed base in that community to respond like I talked about with those grouse forbs and more succulent forbs without seeding. The only plots that got seeded in that experiment were our Dixie plots, but it was a drought year that first year post-treatment, that first growing season post-treatment, and we saw no response from our seeded species. So we basically, it's as if we never seeded, and that stayed true through the rest of our vegetation sampling. On DLL, some of their areas they seeded, some of their areas they didn't. A lot of their, when they dissed sagebrush areas, they reseeded, and they saw a response from their seed, and grouse were using those seeded areas. So, but this was at lower elevations in that basin big sage, and I think there may be some appropriate seeding that you could do there.
Well, great. Thank you all, and thanks to Dr. Dahlgren for your time and your expertise. Pete's on the topic. That brings us to the end of our call here. I'll just take a second to plug Utah State's upcoming conference in Salt Lake City, February 23rd through 26th. Sagebrush Ecosystem Conference, all hands, all lands. I know there will be at least half a dozen or so SGI folks in attendance, and so that might be a great opportunity to follow up. I know Dr. Dahlgren will probably be pretty busy running around, but maybe a chance to grab him in the hallway or at a social and pick his brain a little bit more. So, and if you're not registered for that meeting to be there in person, you can also view it remotely, which I know a number of you guys are already doing. So if that interests you, it looks like it's going to be a good conference, lots of technical resources, and so feel free to look into that between now and, again, the 23rd through 26th of February. So with that, Dr. Dahlgren, thanks so much. I think that was really valuable, and we'll talk to you all later. Thanks.
Thank you.